Respiration is an essential reaction carried out in the cells of all living organisms. It is used to transfer energy from food, so it can be used for life processes such as building new molecules.
Respiration can be aerobic, which involves oxygen, or anaerobic, which takes place without oxygen. In this pod, we will be outlining how to investigate how temperature affects aerobic respiration. The word equation for aerobic respiration is glucose plus oxygen forms carbon dioxide and water. You can measure the rate of respiration carried out by living organisms by either measuring the volume of carbon dioxide produced or measuring the volume of oxygen used.
You can investigate respiration in yeast by capturing yeast cells in small beads of a gel-like substance called calcium alginate. The yeast can survive inside the beads, but they are immobilized, meaning they cannot move out of the beads. This is a useful technique in industry, where yeast is used in fermentation to produce alcohol. You can produce and use immobilized yeast in the classroom to study the rate of respiration.
To prepare the alginate beads, you first rehydrate some dried yeast by mixing it with water and leaving for 10 minutes. Then you add alginate solution to the yeast mixture and stir. To make the beads, draw up some of the mixture into a syringe and add drops to a calcium chloride solution. The calcium ions form cross-links in the alginate to form gel-like beads that contain the yeast cells. The beads are removed from the solution using a tea strainer. You now have immobilized yeast to use in your investigation.
This is how to measure the volume of carbon dioxide produced. Place the yeast in a sugar solution inside a conical flask with a bung to which a delivery tube has been fitted. The other end of the delivery tube is placed under an upside-down measuring cylinder full of water. You need to make sure that the bung is tightly fitted at the top of the conical flask so no gas can escape around it. The yeast will carry out respiration and produce carbon dioxide. This will bubble up through the sugar solution and into the delivery tube before entering the measuring cylinder. The volume of carbon dioxide can then be measured.
To investigate how temperature affects the rate of respiration, the conical flask can be placed in an electric water bath at different temperatures. There are safety issues surrounding the use of electric water baths. Be careful not to touch the controls with wet hands, and make sure that you use test tube holders when removing glassware that is hot, so as to avoid burns. You can draw a line graph of your results by plotting volume of carbon dioxide produced on the y-axis of your graph and time on the x-axis.
The rate of respiration is represented by the gradient of the lines. Using the results shown here, an example is that at 30 degrees Celsius, the volume of carbon dioxide produced after 60 seconds was 40 centimeters cubed. The gradient is 40 divided by 60 equals 0.7 centimeters cubed per second.